What does it take for a cell to make a copy of itself? And what happens if there's a mistake? That's what we'll be discussing in this module. So this information will be coming from chapters 6 and 7 from our textbook. And the main goals of this module is for learners to be able to describe the life cycle of a cell, explain the differences between the different types of cell division, and to recognize the cause and difficulty in treating cancers. So there will be 3 lecture videos for this module. The first will be the life cycle of the cell, then 2 types of cell division, and lastly, cancer and errors during cell division. So the cell is the basic unit of life. Cells are also finite. They normally have a limited lifespan. A single cell will only live for so long. Now, prior to that cell dying, it may instead divide to form 2 daughter cells. In this way, life continues from one cellular generation to another. So what is the significance of cell division? Well, for a unicellular organism, by dividing into daughter cells, it has reproduced. The 2 daughter cells have resulted from a single parent cell. In bacteria, this form of reproduction is known as binary fission. And is the simplest form of cell division. Now, when we're talking about a multicellular eukaryotic organism, like us as humans, cell division is important for reproduction, but it's also essential for renewal, repair of tissues, and growth, and even of our full organism. We are larger now than we were when we were born because of cell division. We have all had injuries, cuts, and scrapes on our skin that have healed because of cell division. The division of a eukaryotic cell is more complex than that of a prokaryotic cell because eukaryotic cells are more complex than prokaryotic cells. In this module, we will discuss 2 different forms of eukaryotic cell division. Now, for a cell to divide, it needs to go through the cell cycle. And this cell cycle is a series of steps. There are 2 main stages to the cell cycle: interphase and cell division. And both of these, in turn, can be broken down into smaller steps. So interphase is the time that the cell needs to prepare for cell division. A cell prepares for division by increasing in size, increasing in protein content, and most importantly, by copying its DNA. Interphase is made up of 3 main stages. The G1 phase is a time of cell growth and preparation for the copying of the DNA, which takes place in the next step, and that is the S phase. The S phase is when the cell copies all of its DNA, all of its chromosomes and genetic material. And the S in S phase stands for synthesis, because that's when all of the DNA is being copied. Following the S phase, there's the G2 phase, which is a second period of growth after copying the DNA. Yet this happens before the steps of cell division. So interphase is made up of these 3 phases, G1, S phase, and G2, when a cell is actively dividing. But if the cell is not preparing for division, If the cell is alive, it's doing its functions, but it's not actively preparing for division, it's actually in a step called the G0 phase. So the G0 phase, it's not technically a stage of interphase, but the G0 phase is when a cell is alive, performing its functions, but it is not preparing for cell division. Some cells will stay in G0 phase for days, months, or even decades. And it is possible that some cells will not divide again. This is true for many of the neurons that are established within our brain. Copying the DNA is important before a cell can divide. So what is the DNA used for? Well, the DNA is the most important source of information that a cell needs in order to function. DNA determines which proteins are made in a cell, how much of those proteins are made, and when those proteins are made. So a cell must have its DNA to be alive and to function. Before a parent cell divides, it copies all of its chromosomes. A chromosome is a complex of DNA and protein. Now, the DNA is the information, and it's a long, thin, thread-like molecule. The proteins are there to help organize the thread. If you are familiar with a seamstress, or if you sew yourself, or maybe even wiring and an electrician, if you have a long thread or a long wire, the best way to keep it organized is to have it wrapped around a spool. Well, the proteins are sort of like the spool, and the DNA is like the thread. The proteins are there as scaffolding to keep the DNA organized and contained. Prokaryotic cells have a single chromosome. So one structure, which is a loop of DNA and its associated proteins, whereas certain eukaryotic cells can have over 100 chromosomes, potentially. A typical human cell has 46 chromosomes. After the S phase, the DNA has been duplicated. It has been copied. These duplicated chromosomes then need to be divided between the two new cells formed by cell division. And the two resulting daughter cells are genetically identical to each other and genetically identical to that original parent cell. When we look at DNA, DNA molecules are enormously long. The DNA of a single human cell, if you were to unwind it, to take it off the scaffolding and stretch it out, lay it out end to end, the DNA of a single human cell is nearly 2 meters in length. That's taller than the average adult human. And that's just from a single cell. If we were to take all of the DNA from a single human and stretch it out end to end, you would likely have approximately 133 astronomical units in length. What's an astronomical unit? That's the distance between the sun and the Earth. So it's not just that the DNA could wrap around the equator, not just that the DNA could go from the Earth to the moon, not just that the DNA could go from the Earth to the sun. 133 astronomical units is sort of the diameter of the orbit of some of the outer planets of our solar system. So an incredible length of DNA, all of that packaged and contained within the cells of your body. So as I had mentioned, DNA is tightly packaged with proteins. Those proteins help keep the DNA organized, and we call that resulting structure a chromosome. A chromosome is composed of one double-stranded molecule of DNA and its associated proteins. So a chromosome is very long and thin, like a thread. As we know, if the goal is to keep a long and thin thread organized, the easiest way to do this is with a spool. And that's the function that the proteins serve in this structure. It turns out that there are different types and different reasons for cell division. We'll discuss that in our next video.